Hello and welcome to my presentation on inherited disease. I am Hannah Lilly. So our DNA is found in the nucleus of our cells. It carries our genetic information. This is information that's unique to us: eye colour, height and other biological characteristics. Our DNA provides instructions for making proteins which control cell structure, function and chemical reactions in the body. It is inherited from our parents and passed to our offspring. It helps growth and control and development in the cell. But how does it do this? Through protein synthesis. Each new cell produced needs its own copy of DNA. mRNA, the messenger DNA, plays a key role in protein synthesis. It carries genetic instructions. Polymerase copies sections of DNA by finding the nucleotides. This then creates new instructions that go over to our ribosome. Our ribosome is a protein-making machine of the cell. They read mRNA 3 codons at a time, joining amino acids together in order to make a protein. tRNA, see a drawing here, help this process by translating the mRNA message into a correctly assembled protein. They bring in the correct amino acid joined by a peptide bond, and this eventually forms an amino acid chain for the protein. But if all cells contain all DNA information, how does each cell know which part to translate? How do your eye cells know not to produce stomach cells? The portions that are red need to be controlled. The right proteins must be made at the right time through a process called gene regulation. RNA, for transcription to take place, the enzyme that synthesizes RNA polymerase must attach to the DNA near a gene. So here's my section of DNA. Part of it is the gene, and this is polymerase at the start of the section that needs to be transcribed. RNA polymerase binds to the promoter region and then builds the mRNA using complementary base pairs. This is when polymerase reaches the stop codon in the gene, this terminates the process as the mRNA strand is complete. Not all bases in the DNA code for amino acids, so the mRNA just transcribed contains non-coding regions known as introns. Introns are removed with the help of enzymes before mRNA leaves the nucleus, leaving just coding regions called exons. If at any point this instructions data is incorrect, that can create a point mutation. A point mutation is a changing in structure of a gene, bases G, C, A or T, that affect the protein, resulting in the change in the characteristic, which is passed to further generations. It can be spontaneous as a result of badly copied DNA, like we've just seen, or it can be induced, caused by external factors that damage DNA, like smoking. Effects of this is almost always harmful. It can change one or more proteins, and proteins could end up being non-functional. However, there are benefits too, such as evolution. Here you can see the correct DNA sequence travelling over to our ribosome, being read by our tRNA that brings in the correct amino acid. However, if one of these bases was incorrect, that can create a mutated DNA. travels to our ribosome, brings in an incorrect amino. Further possible mutations are, like, the best possible one is called a silent mutation, where a similar codon for the correct amino acid outcome is the same. So for example, in our first amino acid sequence, we've got GAA. In our second, however, incorrectly, we've got GAG. If you have a look at the table of amino acids to mRNA bases though, GAA and GAG belong to the same amino acid, glutamic acid. Missense mutation, which is a similar mistake, however, each amino is different, resulting in the wrong amino acid being used. Frameshift mutation is a result of insertion or deletion, making the rest of the sequence incorrect. So here you can see the first ones match, however after that the remaining sequence is incorrect. Nonsense mutation is where a codon is changed for the stop codon, cutting the protein sequence short. Cystic fibrosis is an inherited genetic disorder. It is caused by a mutation in the CFTR gene, and it is an inherited disease because the mutation is passed down from parents to children through recessive inheritance. You see here, I've got Punnett square displaying the father having a cystic fibrosis gene and the mother both having a cystic fibrosis gene. This results in a 25% chance of the child having cystic fibrosis. People with cystic fibrosis have a faulty CFTR gene which is found in chromosome 7, over here, chromosome 7 with the faulty gene. After transcription translation, that can create a faulty CFTR protein. The CFTR protein is responsible for the control of movement of salt and water in the cells. The genes that produce the proteins that are responsible for this do not perform their job properly, resulting in thick mucus in the lungs. So here you can see a healthy lung cell, our healthy CFTR protein doing its job, letting these chloride ions pass through salt and water. Whereas in the faulty gene, creates a faulty CFTR protein, which is stopping these chlorides getting through, resulting in a thick mucus lining. This can present in symptoms in patients such as persistent cough, mucus, and shortness of breath. Human gender is determined by the sex chromosome, and we receive 23 chromosomes from each of our parents through the gamete cells, so the egg and the sperm cells. The mother always contributes 23 X chromosomes in her egg cells. The father contributes either X or Y chromosomes in his sperm cells. Therefore, the father determines the biological sex of the child. This is demonstrated in my Punnett table, here showing a 50-50% chance. A sex-linked condition is caused by a gene located on a sex chromosome, most commonly the X chromosome. Because males only have one X chromosome, a single recessive faulty gene on the X chromosome will cause the condition. Females have two X chromosomes, so a normal gene on the X can make a faulty one. For example, colour blindness, haemophilia, which you generally see more in the father. So in this case, if you have an unaffected father and the mother is a carrier, the daughters are unaffected. The gene is shown in the little h, whereas the sons have a 50-50% chance of being affected. So therefore, males are more likely to be affected. Some inherited diseases can be as a result of chromosomal abnormalities, where there is a change in the number or structure of chromosomes. These abnormalities can disrupt genes and lead to inherited disease if they are passed from parent to offspring, resulting in developmental and health problems. Here you can see some structural abnormalities with the chromosomes, such as deletion, where a section is completely missed out, or duplication, where a section is actually copied. Numerical abnormalities is we should receive 23 chromosomes from our mum, 23 chromosomes from our dad. If this is, however, an abnormal amount, so we receive more or less, that can cause diseases such as Down syndrome is caused by an extra copy of chromosome number 21. Genetic screening is a process used to identify individuals who may carry genes or chromosomes associated with inherited disorders, often before symptoms appear. It involves testing DNA for faulty genes or chromosomes, uses biological samples that are lab tested. Results are then explained through genetic counselling, and then the individuals are helped to identify any risk of inherited disorders. DNA recombinant technology involves combining DNA from two different organisms to produce useful proteins or traits. It is widely used in medicine, agriculture, and industry. DNA is cut, joined, and inserted into host cells like bacteria, in my example, for replication and large-scale production. In my example here, the gene responsible for producing insulin has been cut from a chromosome of a normal insulin-producing human and inserted into a plasmid and sealed with enzymes. This created gene is then inserted into suitable bacterium, which then replicate, producing more insulin genes. This insulin is then extracted and used to treat people with diabetes. Recombinant DNA technology raises moral and ethical issues because it involves altering the genetic material of living organisms. Some people are concerned about safety and long-term effects on human health and the environment, including the spread of genetically modified organisms. There are also ethical concerns about animal welfare, fairness in access to genetic treatment, and the possibility of human genetic modification being used for enhancement rather than medical treatment. Do patients fully understand genetic treatment? While recombinant DNA can treat disease, ethical concerns are raised.